Our lab focuses on the role of epigenetic signatures in rare disease.
Heidelberg Epignostics has established methylation signatures and genomic biomarkers to precisely classify brain tumors.
The genetic and environmental etiology of disease can be complex.
Our users require a streamlined, cost-efficient workflow for reliable turnaround times.
We need to systematically collect high-accuracy methylation and genomic data to deepen our understanding.
At Illumina, we're listening to you and relentlessly innovating for you.
We see that you need a more efficient way to study the genome and methylome.
With the Illumina 5-base solution, now you can detect genomic variants and methylation events simultaneously with exceptional accuracy, simplicity, and scale.
Most methylation detection methods come with trade-offs. They reduce nucleotide diversity, damage DNA, and they leave gaps in your data.
The Illumina 5-base solution takes a different approach.
Our proprietary enzyme converts 5-methylcytosine to thymine in a single-step base conversion while preserving DNA integrity and library complexity.
This unique capability enables clear, accurate results and helps you get the most from every sequencing read.
The Illumina 5-base solution is available in 2 versions: a whole genome assay, Illumina 5-base DNA prep, and a targeted assay, Illumina 5-base DNA prep with enrichment. This solution is already transforming research to allow you to focus on results.
The Illumina 5-base solution is transformative.
It's indeed a game changer. The dual insight maximizes information out of a single sample.
We are uncovering DNA variants and methylation signatures linked to disease, data we simply couldn't see in a single run before.
The dual detection capability is a huge leap forward.
It is very exciting to see the high level of specificity with the 5-base data.
It successfully classified diverse brain tumor types and reported key biomarkers, all in one streamlined workflow. The 5-base solution is scalable, works on our current Illumina platform, is efficient, and provides deeper insights.
We strive to create real transformation through innovation. Making multiomics more accessible gives you the tools to shape the future of biology.
Integrate multidimensional insights with your current NGS workflow today.